Recording and dive into it. So for this week, we will be talking about bacteria again in the context of its the way in which it gets its nutrition from the environment, as well as a little bit of the ecology, so where different bacteria live, and then their growth. What's their patterns of growth? Do they form biofilms? Do they what are the different stages of growth? And then that'll wrap it up for chapter 7. So we'll see what we can get done in the next 2 lectures. Here's looking at an x-ray of the lungs of a patient with cystic fibrosis. And so it makes patients with cystic fibrosis are susceptible to lung infections, one of which is Pseudomonas, and it's able to then use its fimbriae, which you remember is one of those outer structures used for attachment, and it'll take residence in the lungs. And as you guys hopefully remember from several lectures ago, the lung is an area that should be sterile, right? And if it's not sterile, then you have an infection. It's not like the gut. It's one of those tissues. Anybody, what's another tissue that's supposed to be sterile? Something else.
Liver.
Liver. Anything else? Yeah.
Bladder.
Bladder. Right. Exactly. Anything else?
Pretty much everything except for our digestive tract.
Yeah. So, you know, once you start, yeah, and blood, right? If your blood gets infected, then you're septic. So, yeah. But yeah, as you said, pretty much anything that is not from your digestive system, your GI tract, it should be free of bugs. So in the case of patients with cystic fibrosis, they, like I said, they're more susceptible to these infections. The chapter itself is going to be looking at, like I said, how different microbes extract nutrients from the environment. And then whether that's from the soil or from the ocean or from the hosts they're infecting, they all have different strategies to do that. Some are photosynthetic, some are not. Also, how do they get their energy, right? So again, some can be photosynthetic. They can derive energy from the sun. They can derive sugars from the sun. And so we'll talk a little bit, just kind of surface level things about how microbes get the energy that they need to grow and to make new proteins and to move, things like that. We'll also talk about how things get in and out of the membrane, because remember, these membranes for bacterial cells are semi-permeable, so they selectively let things in. And that can be done in a passive way or an active way. And we'll talk about those two methods in this chapter. We'll also talk about the different niches that microbes can thrive in, and how we've mentioned in the past extremophiles, things that thermophiles, halophiles. We'll talk about that in a little bit more detail in this chapter. And then the different names that we give to the different bacterial or microbial organisms that can thrive in areas that are typically inhospitable to most life. And then a little bit about how microbes can form communities, right? And so we talked about biofilms, this production of a network of bacterial cells that can then do what's called quorum sensing, which is a way to basically communicate from one cell to another about things that are happening in the environment, whether they need to take in nutrients or whether they need to release toxins. It's a communication system. And then finally, we'll talk a little bit about just overall growth and the process of cell division and just properties of their growth phases. So there are going to be points where they're not dividing a lot. There's going to be points where they divide a lot again, and then there's going to be points where that levels off, and then cells die. So each of these stages have a certain name to them, and we'll go through that. Okay, any questions so far? That's just kind of the outline of this chapter. Okay. All living things have to get nutrients, whether it's humans or plants or microbes. You know, if it's a living thing made up of cells, it has to attain things from the environment to keep that cell carrying out its functions. The cells are great at making a lot of things that they need, but they can't make everything they need. And so certain elements we term bioelements because they are required for life. So carbon, hydrogen, oxygen, phosphorus, potassium, nitrogen, sulfur, calcium, iron, sodium, chlorine, magnesium, all of these are bioelements. Okay, your nucleic acid is going to be a combination of carbons, hydrogens, oxygens, and nitrogens. Sulfur is going to be used for the disulfide bonds to link things together, to give them secondary structure, tertiary structure. Calcium is required for to let things in. So calcium ion channels, all kinds of things. Magnesium is required for DNA replication. All of these things are necessary and they come from the environment. Okay, right? Living things don't make elements. Elements are already made. They take it in from the environment and they use it to make the biomolecules that they use. Essential nutrients. These are things that the organism must have from outside of its cells. And so these can be macronutrients like proteins and carbohydrates. They can also be micronutrients, manganese, zinc, nickel. Okay. And just as these things are all required for us humans, bacterial cells do the same thing. And this goes back to what I said last week, in that because evolution doesn't reinvent the wheel, it doesn't start over unless there's some driving force for that. And so the same cellular processes that occur in us really were just recycled from earlier microbial cells. Organic nutrients are going to be nutrients that are carbons and hydrogens. So those of you that are taking organic chemistry right now, you know that that's what organic means, the presence of carbons and hydrogens or hydrocarbon bonds. And so things like methane, that's the simplest organic compound. It's just a carbon and 4 hydrogens attached. But then it can get more complicated. You can get carbohydrates, lipids, proteins, nucleic acids. Now we're going to take these hydrocarbons and we're going to make bigger molecules with them. And we're going to tack on nitrogens and oxygens. And so these are the molecules of life. These are the things that make up the tissue or the cellular structures the proteins of these microbial cells and our tissues of us humans, right? Inorganic nutrients are going to be things that are still important, but aren't that hydrocarbon bond, right? So things like salts and metals, gases, those come together and they help out the organic nutrients. Okay. When we look at the makeup of a cell, 70% is water. And again, these are things that are universal, right? I could be talking about a microbial cell right now. I could be talking about your cell. 97% of the dry cell weight, so not the cytoplasm and stuff like that, just the biomolecules, if you were able to just extract all of those, the vast majority of it is proteins, because proteins are the workhorses of the cell. They do all the communications, they do all the functions. And so the majority of them are going to be, the majority of the components in a cell is protein. And then also the vast majority of the cell is made up of these essential elements, these bioelements, carbon, hydrogen, oxygen. That makes up DNA and protein, right? And if most of the dry cell weight is protein, then most of the bioelements that go into making the protein are going to be there. So carbon, hydrogen, oxygen, nitrogen, and then things like phosphorus also for the nucleic acid, and sulfur for these disulfide bonds. Okay. Now, there are two different ways that microbes can get their nutrition. And if they are heterotroph, that means they get carbon, which is that essential bioelement, in some kind of organic form, whether it's proteins, carbohydrates, or lipids. And they get that from other living organisms, okay? And that could be from live organisms, like a host, or it could be from organisms that have expired and then now they're recycling those carbohydrates and lipids and proteins. The autotroph is going to be different. It's not going to get its bioelements from other living things, okay? It's going to utilize things like carbon dioxide to get its carbon, rather than proteins and carbohydrates and lipids, which all also contain carbon. It gets it from the CO2, and it's going to do that, via photosynthesis. So autotroph, auto means, self. It doesn't need some other living thing to get it. So be familiar with those two different types. So again, for carbon, you have autotrophs, heterotrophs, energy source. Now we're not talking about getting your carbon to make up your biomolecules. We're talking about where you get your energy, chemotroph and phototroph. So a chemotroph is going to get energy from chemical compounds, those compounds that come from living matter, really, or recently expired matter from a previously living organism. Whereas phototroph, photo, light, right, it's going to get its energy through photosynthesis. Okay, so this is how we break down the major different types of microbial carbon sources and energy sources. And then you can start blending them together and say, okay, what is a photoautotroph? Well, a photoautotroph is going to use photosynthesis to get its carbon, right? So things like plants and algae and cyanobacteria, those are examples of photoautotrophs, and they're oxygenic. Why are they oxygenic?
Because they use oxygen in their systems.
Well, they produce oxygen. So remember that photosynthesis is using light and carbon dioxide to make sugar, and what's released is oxygen. So oxygenic means that oxygen is a byproduct of their metabolism. Okay. There are photoautotrophs that are anoxygenic, meaning they do photosynthesis, but they are unusual characters in that they don't really produce oxygen as a byproduct. Instead, they produce sulfur. Okay. And they use this special type of bacteriochlorophyll to do that. So plants, algae, cyanobacteria have typical chlorophyll that we think of, that we've learned about in botany, right, with photosynthesis. But these anoxygenic ones use their own form of bacteriochlorophyll. And that can give them a purple or a green pigment, depending on the particular type of bacteriochlorophyll. You also have chemoautotrophs. Okay, so chemoautotrophs, they're autotrophic, meaning they don't need other living things to get their carbon. They can get it from inorganic substances that contain carbon. Not all organic things are, well, I should say, the only source of carbon, organic living things aren't the only source of carbon, okay? And like I said, methane is an organic molecule, not considered organic in that it's living. A little bit of semantics there that can be confusing because it's used interchangeably. But methanogens in particular, those are going to be chemoautotrophs that produce methane under their conditions. Okay, so if we go back here to oxygenic and anoxygenic, that's producing oxygen or not producing oxygen. Here, methanogenic would be, instead of producing oxygen, you're producing methane. Okay, and here are some examples of that to the right. Organisms that are methanogens. All right. So when we're talking about heterotrophs, now remember, what are heterotrophs? Those are things that rely on other living things to get their source of carbon. The majority are going to be chemoorganotrophs. Wow, now we're really slinging some fancy words here, but a chemoorganotroph is going to get its carbon and energy from organic compounds. Okay. And more likely than not, it's going to have aerobic respiration. So it's going to be requiring oxygen. When you have these microbes that live in areas where there isn't oxygen or there's low oxygen, then you start having like methanogen type microbes. Okay, so that's an anaerobic condition. Whereas here, the vast majority of heterotrophs, because they rely on living tissue of some sort, I hate to say tissue, but living matter, and most matter that's alive requires oxygen. So most of these things are going to be in aerobic conditions. And so we can break this up into two different categories. Saprobes are free living microorganisms that feed on organic detritus. So just dead mass, biomass. So maybe they're breaking down fruit that has fallen off of the tree. and is now just starting to decompose. Why is that decomposing? It's because the bacteria have sought that out and are feeding on all the sugars in that fruit to get their carbon and their energy. Okay. A lot of fungi are saprobes, and they actually help the biodegradation process, right? If we didn't have these microbes breaking down leaf litter and breaking down just animals that die out in the wild of natural or unnatural causes, all of this stuff would just pile up. It would take much longer for that to decompose. But thanks to microbes, they speed that process up. And these are often going to be opportunistic pathogens or facultative parasites. And what does that mean? It means both of those things don't have to have a host. Because remember, saprobes, as we say, are free living. And what do we say an opportunistic pathogen was on the quiz a couple weeks ago? It's something that doesn't need to have a host. But if that host is for whatever reason compromised, immunocompromised, then it allows that microbe to take up residency. But it can go either way. Facultative parasite, same thing. Facultative means it's an option, okay? Obligate or obligative means there is no choice. So saprobes can be free living. If they come across a host and it's the right conditions, they can jump into that host. But most of them don't do that. Parasites, on the other hand, that's the other chemoorganotroph. Those have to have a host. And they get their carbon and their energy directly from the host, okay? Not from breaking down dead organic matter. And these are most likely going to be pathogenic because, again, they are infecting the host. And when you are deriving your carbon from the host, well, you must be breaking down that host's biomolecules in the process. And unless you're just taking things that have been recycled, it usually means that you're breaking down the host's proteins or breaking down the host's cell wall and extracting those things, okay? And that's going to cause disease in some form. Okay, any questions so far on this? A lot of little keywords here, but be familiar with these different things. No, we're good? Okay. The cell wall is the protective coat for a microbe, but it has to be semi-permeable so that things can get out, like waste or toxins to facilitate infection, or just signaling molecules when it's quorum sensing that we'll talk about in the biofilm. Things need to get out. Things need to get in, right? So when we talk about these heterotrophs getting their energy source from external environments, they have to somehow break down that organic material so that it's smaller and can get across the membrane. So what they do is they excrete enzymes, and those enzymes help break down that dead fruit, that rotting fruit, that leaf litter in the soil. Or, if it's pathogenic, then the host's cellular materials. Breaks it down into smaller components that then can be taken in by that cell wall. Okay. So it releases the enzymes. The enzymes then start to hydrolyze the bonds, or what they do is they break down the bonds that hold this material together. And so that now they're reduced to their parts, not the sum, And those little parts can now be transported back into the cell and utilized to make proteins for the bacteria. Concept check. If an organism is degrading large organic molecules to get its carbon and energy, which one of these things would it be?
C.
Exactly. So photoheterotroph. Photo means it's getting energy from the sun. So that immediately gets rid of A and B. And D, chemo, okay, well, that means, it's getting it from, it's chemically derived. It's not derived from the sun, but it's an autotroph. So it's not going to be degrading other things. It's going to be getting it from like carbon dioxide, like we said. Okay, so the only one that really fits here is the chemoorganotroph. Good. Now, as I said, there are different mechanisms to allow things to come into the cell and to come out of the cell. One way that things are brought in or out is through a process called passive transport. And as the name implies, it's the transportation of things passively. It doesn't require energy to make that happen. It's somehow recognized as favorable and allowed to come in. And oftentimes this is done by three different ways. One is just diffusion. One can be osmosis. So when concentrations are different on the outside of the cell versus the inside of the cell, water will move one way or the other, and things can be brought in with that water transport. And then facilitated diffusion is going to be particles that need some kind of carrier, something to deliver it. And we'll talk about these now. I guess not now. First, we'll talk about the other. Well, I'll give you an overview of the other form. The other form is active transport. So whereas passive doesn't require energy, active transport does. It also needs a carrier protein. Okay. And so this can be, again, three different ways. It can be carrier-mediated active transport, which we'll talk about. It can be group translocation, which we'll talk about. And then bulk transport, which is endocytosis, exocytosis, and pinocytosis. Those are just ways that the cell membrane engulfs something. And we talked about that a little bit when we were going over the immune system and how things like macrophages and other phagocytes engulf things. Similar process with these bacterial cells. Okay. Molecules are going to, in a passive way, move with the concentration gradient. Okay. And that is going to be one way. So if something is more concentrated, it will potentially move that direction, or depending on the substance that it's trying to move, it'll move away from that to a less concentrated state. There's also this concept of just random thermal movement. Okay. Now, what is thermal movement? Thermal is heat. Heat is energy, and energy is anything that has the capacity to do work, right? And so things can move when you heat them up. I think this is a cup and it has a sugar cube in it, and it's showing that if that, it's really, really like watered down coffee or coffee with a bunch of milk in it or something. And if that is hot, it's going to help break and dissolve that sugar more quickly than if it was cold. Osmosis is when we have these membranes of the cell and they don't let certain things get in. But they do allow diffusion of water. And again, that's passive, okay? But when the concentrations of these larger molecules are different on one side of the cell versus the other, there will be a movement of water in a certain direction until equilibrium is achieved. And these are the three different types, okay? So you have isotonic, hypotonic, and hypertonic. And if you guys have had any kind of cell biology, you'll be familiar with this. But essentially, what's going on if something is isotonic, iso means the same. And so the concentration on the outside of the cell is the same as the concentration on the inside of the cell. And so you have a homeostasis in a way of the water. Water, the same amount of water that's moving out of the cell is the same amount that's coming in. And so it's isotonic, it's balanced. If things are hypotonic, now what's going on? Now the concentration is such that water is moving in, okay? And you're getting, this can usually lead to, if it's, so for instance, why are things cured? I mean, like jerky or like dried fruit and stuff like that. Why do you salt meat to make it into a jerky?
When you salt stuff, you make the solution so hypertonic that it sucks all the water out of pathogenic microbes like bacteria and stuff, and they just dry up, and then you can't infect it.
Right. So, I think it's the reverse. I think it's, you make it hypotonic and then it explodes the cell. Right. So if you give it a bunch of salt, and the salt is an electrolyte, right? So when we are dehydrated, we drink Gatorade or something that has salt, and that allows water to go in. Okay. But if too much water goes in, the cell will explode. And so if you just put a bunch of salt on meat, for instance, it will create this hypotonicity and the cell will explode. And then hypertonic is like what you mentioned, the water leaves the cell and it becomes dehydrated. And so then all the water's gone. Cells have to be 70% water. And so as you can imagine, that's detrimental to the cell. Okay.
Are you sure that's not backwards? Because if all the salt is on the outside of the cell, it's going to be super high concentration outside of the cell. That's going to pull water out. But if you have something where it's just pure water and all the concentration is inside the cell, that's going to make water want to go inside the cell, and that's what will make it explode.
So with the salt on the outside, right, the salt is going to be super concentrated on the outside.
Right, and that's going to want water. That's going to create an area of extremely high osmotic pressure, and that's going to make all the water want to leave the cell, and it'll dry up.
No.
The water wants towards the area of high concentration.
No. So like if you look at this diagram here, it's showing highly concentrated solute on the outside of the cell. Okay. And so as a result, the water is going into the cell. Whereas here, it's not.
That doesn't make sense though, because if you put a cell in just salt, where's the water going to come from to make the cell explode? If you just put something in pure salt and there's no water, there's no water that's going to go into the cell to make it explode. All the water is going to come out of the cell.
Isn't osmosis, osmosis goes to the higher concentration as well, not the lower, right? So I'm seeing things.
No, yeah, this is good. So I see your points. Yeah, that makes sense. So if without water, so what you're saying is there's no water on the outside, it's just salt. And so there's no water to go into the cell to blow it up, right?
Yeah.
Yeah.
Yeah. So if you were to just inject like distilled water into somebody's blood, you want to put isotonic solutions into people's blood so that it doesn't change the concentrate, or like doesn't change the water level inside of their erythrocytes, right? But if you inject them with pure distilled water, all that water that's in the bloodstream, they're going to try and your blood cells are going to try and like balance the concentrations, and all those extra water molecules are going to go into your blood cells, and then they'll explode.
Right. Right.
That would be a hypotonic solution, because distilled water is less concentrated than blood.
Yeah, no, you guys are right. Thank you. And someone had mentioned, the whole concept of osmosis, and it's traveling to the higher concentrations. So, okay, yeah, thank you. See, this is great. This is what I like about this feedback, because, that makes sense, and I got that wrong. So, yes, if you salt beef jerky, then that would make the cell dry up. Yeah. Okay. Cool. Thank you, guys. Any other questions on this or comments? No? Okay. Be familiar with these 3 different types. I won't ask you about how to make beef jerky, but know what the difference between iso, hypo, and hyper is, okay? In terms of the movement of water, where it's going, inside the cell, outside the cell, or if there's a balance between the two, okay? All right. Facilitated diffusion. So this is also going to be a form of passive transport, but there has to be some kind of carrier protein. And in this case, it's this green structure embedded into the cell membrane. And then above that, where all these particles are, that's going to be outside the cell. Below is inside the cell. And so these different carrier proteins have a specificity to certain types of molecules. So a lock and key kind of scenario. And so once that particular solute binds, it recognizes it, and then it allows it to migrate through the cell into the inside of the cell. Okay. Of course, this is limited by the number of sites on that carrier protein. So sometimes it just has one site. And so that's going to slow the rate of transport. Or it can have multiple different sites, which will, of course, have a slightly faster rate. Things could be balanced, though. If you had fewer carrier proteins that had multiple sites than you did of carrier proteins that just had one site, it might be about the same. So it really just depends on how many carrier proteins are embedded into the membrane, and then also how many sites are on that carrier protein to allow the particles through. Carrier-mediated active transport. So now with active transport, we have to have energy put into the system. And so these are going to be transporter proteins that are work like pumps, okay? And so you have the solute on the outside. It will bind with the transport protein, and then via ATP, that will allow this to shuttle it through the cell into the inside of the bacteria. Okay. So the ATP is what produces the energy to allow the pump to work. And so this is going to be important for things like sugars and metals and sodium that can't just passively diffuse inside. Okay. because you got to be able to regulate that to some degree. And then in-group translocation, this is going to be where something from the outside is bound to the carrier protein and then modified to a certain extent on its way in, so that you have something that's ready to go. Once it enters, it has its modification. And this can conserve energy because now you're only using energy to pass it through. In the process, it gets modified. So it doesn't need to be modified in a second step, which could potentially require more ATP for that modification. Okay, so it's a convenient way to bring in certain certain elements or biomolecules and modifying them so that they're ready to go. In-group translocation. So the difference here was with the carrier-mediated active transport, it's not modifying those molecules per se. Okay. And then endocytosis, that's the last one. That's where they engulf a particle. If sometimes it's too big to go through these pumps. And so they create, in doing so, they create like a little vesicle or a phagosome. And they use these structures called pseudopods, pseudopod meaning kind of like a foot, not really, to do that with phagocytosis. Or if it's penocytosis, it's going to just gulp in a bunch of fluid or dissolved things using microvilli, right? And so this is the illustration looking at that. For phagocytosis, you have the solute bind to the cell, or at least encounters the cell, and that triggers the elongation of these pseudopods that will then come up and surround it and bring it into a vacuole or a vesicle, which can then open up and release that material. With penocytosis, you're going to use the microvilli on a cell, bacterial cell, and it's going to just engulf this droplet of water, or if it's in a watery environment, it'll just engulf some water and bring it in. Or in this case, it's oil droplets, and then that way it's internalized whatever is in that water to use for protein synthesis. Okay. All right, so we'll do this concept check and then we'll wrap it up for today. If a cell is concentrated glucose solution, and its glucose is moving into the cell via a carrier protein, what do you think is the example for this?
Facilitated diffusion.
Facilitated diffusion? Facilitated diffusion. Makes sense, right? Because we have a concentration gradient and we have it moving in through a carrier protein and no mention of any kind of energy. Okay. Diffusion, that would be what? That would be without a carrier protein. So facilitated diffusion, because it has that carrier protein, that's facilitating the diffusion in the context of the glucose. Okay. Any questions on that? Okay. Well, we're going to, let's take a 10-minute break, and then we'll start getting into the journal club for today and just discuss it and see what your thoughts were on the paper. And I would like to use the paper as an example of how review articles can be helpful, but what some of their limitations are as well. So we'll take a 10-minute break, And I'll see you guys at 11.